Thank you so much for joining us today. Hello, I am Sai Subramanian. Today I'm joined by my colleague, Dr. Manisha Ray. Seven Bridges is a global biomedical data company which provides end-to-end informatics infrastructure for several disease-focused research initiatives. Manisha and I are program managers here at Seven Bridges, managing one such cloud resource platform called the Cancer Genomics Cloud, in short, CGC. The CGC platform is funded by the National Cancer Institute, and it allows researchers around the world to explore massive public data sets, such as the Cancer Genomics Atlas, TCGA, Clinical Proteomic Tumor Analysis Consortium, CPTAC, and other large omics and imaging data sets, alongside secure and scalable computational resources to analyze them. The National Institutes of Health, NIH, has made several foundational investments in funding basic and translational research, which generates massive amounts of data, as well as data analysis ecosystems or platforms to empower the scientific community to analyze these large data sets. Over the years, the CGC has not only enabled researchers to perform collaborative research, but it has been used as an effective teaching platform for training the next generation of national research workforce. Today, I'm delighted to welcome our guests, Dr. Yuri Gusev and Ms. Kritika Bhuvaneshwar from Georgetown University, who are here with us to share their experiences in using the CGC as a teaching platform for Georgetown University's Masters in Health Informatics and Data Science program. Dr. Gusev and Ms. Bhuvaneshwar are our collaborators and long-term users of the CGC. I would like to request all of you to introduce yourself. Let's start with Dr. Manisha Ray.
Hi, I'm Manisha Ray. I'm a senior program manager at Seven Bridges, and I primarily work on the CGC, and I've worked closely with Yuri and Kritika on developing several courses for teaching bioinformatics using the CGC.
Great, thank you. Dr. Gusev?
Hi, I'm Yuri Gusev. I'm Associate Professor of Bioinformatics at Georgetown University Innovation Center for Biomedical Informatics. I'm also Director of a Master's Degree Program in Health Informatics and Data Science at Georgetown.
Great, thank you. Ms. Bhuvaneshwar?
I'm Kritika. I'm a research instructor faculty for the HIDS program. I'm also a senior bioinformatician at Innovation Center for Biomedical Informatics at Georgetown University. I've been using, along with Dr. Gusev, we've been using the Cloud Seven Bridges platform for both research and teaching. And we've also collaborated with both Manisha and Sai for several, I think a few years now.
Wonderful. Thank you so much, all of you. Okay, so let's begin today's session with a few questions. So can you give us a brief background of the Health Informatics and Data Science program?
Sure. Health Informatics and Data Science program is an accelerated master's degree program at Georgetown University. We started it 2 years ago, 2019. And so far, it's been a successful program, attracting students with mostly computational background from around the globe. And we focusing on teaching students and providing them with hands-on experience in all our courses. And one of our choices to provide this hands-on experience was to use Seven Bridges platform to teach students how to handle big biomedical data on the cloud and how to use different kind of bioinformatic platforms to analyze the data.
Ms. Bhuvaneshwar, do you want to talk about it?
Yeah, so for teaching, we've been working with the Seven Bridges platform since 2019. We initially started working on the RNA-seq and a whole genome sequencing. We teach the students how to do an end-to-end workflow for both RNA-seq and whole genome sequencing during the class, and they also get assigned homeworks for that. And it's been a great experience for them because they are able to learn the concepts of the workflow itself. And so it's much easier to learn, for them to learn by using the Seven Bridges platform.
What aspects of the platform in particular make it suited for teaching your students?
So there were really several reasons why we choose Seven Bridges platform. We have searched for appropriate platform for doing our research for quite some time. When RNA sequencing and whole genome sequencing became very popular research applications, especially in cancer research, we really was looking for cloud solutions, cloud-based solutions for our research program. Later on, we realized that the same approach with cloud-based platform for a very large amount of genomic information could be used for training. And after considering different available commercial and non-profit platforms, we have decided to choose Seven Bridges platform because we found it to be most user-friendly and convenient in usage, and also provide a great opportunity to do reproducible and shareable research online.
Also, I want to add, when you're teaching concepts of sequencing to students who don't know anything much about genomics, it's much easier to start from the basics, and a simple end-to-end workflow like in Seven Bridges is much easier to teach as opposed to starting with a virtual machine or a shell script or any of those installations that we have to teach them first. If we have to teach those first, then it makes the teaching much more complex.
Can you help us understand which elements of the CGC platform do you find the most helpful when you structure your course curriculum, and just a little bit more about why those elements are appropriate for the teaching course?
Right. There are very serious challenges which we face when trying to teach a modern genomic and bioinformatic techniques to graduate students. One of the major challenges is to provide students with a real life experience of how to handle massive amount of genomic data, so-called big biomedical data. We spent quite a few years to develop methodologies which allow us to make it easier for students to understand and get a feel, so to speak, of how to work with this massive amount of data. One of the major challenges to make those data accessible and easy to use for students. The amount of data which we're talking about is, for example, TCGA collection is about 2.5 petabytes of data. Obviously, it's not possible to do it on a local machines, local computers, or local workstations. The cloud solution is the only solution we can use to provide access to this data. TCGA collection in particular is not only a great research tool, but we also found it very useful to use for training. And Seven Bridges have direct access, provides direct access to the TCGA collection, as well as to many other very large massive amount of data in publicly available collection, such as TCRA, for example, the image data collection. And this accessibility of big data, big biomedical data through Seven Bridges platform was one of the major reasons why we choose this platform. And in addition, we also found very useful the collection of tools and pipelines for analyzing the data, which are available through Seven Bridges platform. Those tools are available at the cloud platform, right there where the collection of genomic data is located. And that's allow us to follow the major principle of bringing tools to the data as opposed to bringing data to the tools, which is very impractical when you deal with genomic data. So basically, the Seven Bridges provide us in one shot opportunity to teach students how to handle large, big biomedical data and how to use tools to analyze them on a cloud platform.
I think the Seven Bridges has a nice framework for teaching, because the way we set up the infrastructure is we have one project which we create for each of our students, and then we copy the pipeline, and we teach them how to copy the data as well. So in addition to the instructor demoing the pipeline, the student is also able to run the same pipeline at the same time or after class in their own project, and we as instructors are able to track it because we are also collaborators listed on the project. So it's very easy to not only teach, but also to check if they are running the homeworks correctly or the hands-on portion of the class.
So if I can summarize, it sounds like what makes the CGC beneficial for teaching is that the ability for the instructors and the students to all work on the same thing at the same time through the projects. And that includes the data, the tools, and then you can actually see what analysis they're doing all at the same time.
Yes, that's right.
Great. How have the students responded to being taught bioinformatics using the CGC? What do they say about using the platform?
So the students actually appreciate it quite a bit. I must add that the bioinformatic tools, we didn't talk much about it, by themselves. The pipelines we're using in our research and also using for teaching are quite complex and sophisticated. It's a multi-step workflows which include multiple software packages. And in general, it's a tough topic to teach because it's very complex and require students to have advanced knowledge of genomics and computational approaches for genomics as well. So to make it easier for students, we decided to use 7Bridges collection of tools because they are presented in a very user-friendly graphical interface where students have this low barrier to understand and start using immediately the pipelines, which otherwise would require a lot of additional training. So in this way, we found our experience it makes it easier for us to train students, but also for students' perspective, it's much easier for them to start using, and through usage, through hands-on experience, they much quicker understand all the details about the pipelines and about the computational packages we use for genomic data analysis.
Yeah, I completely agree. So before we do the 7Bridges hands-on session in the classroom, we go over the concepts of what each steps in the pipeline are. And that's actually a very complex and dense topic. And so actually we can see on their faces that it's quite difficult to understand. And then we come to the hands-on session, and then they're able to see the pipeline, the workflow in the 7Bridges platform, and they're able to run them quickly. And it's much easier for them to digest once they see it on the SB platform.
Great. That's one of the, I think, one of the wonderful features of the platform, how it simplifies complicated tools and makes it easy to visualize and stitch together newer kinds of workflows. So that's great feedback. So we are talking about tools. So can you elaborate a little bit more on the different kinds of analyses that you have taught? Like, I know genomics is one, imaging is probably one. Can you elaborate a little bit more on the kinds of analyses? Right.
So right now we are using 7Bridges platform in two different courses. One is Precision Health Informatics. This is a course we developed with Critica together and are running during fall semester every year. And for this course, we utilize 7Bridges to teach students how to process genomic sequencing data, specifically RNA sequencing pipelines and whole genome sequencing pipelines, as well as exome sequencing pipelines. Those three pipelines, as I mentioned, are quite sophisticated and also are very important in a field of genomic. I would call them the core pipelines, the core analysis workflows, which everyone needs to know how to use if they want to work in the genomic field. So RNA sequencing probably our major focus currently, where we spend more time with students. and because we have also additional modules for downstream analysis of data generated on Seven Bridges platform. So the students can not only learn how to process the data, but also how to do additional research after you process the genomic data. For imaging, we also started to use Seven Bridges platform because the modern imaging analysis, biomedical imaging analysis, now moving toward quantitative type of analysis, which pretty much similar to what is happening in genomics. Basically, we're talking about feature extraction workflow, which is implemented on Seven Bridges platform, and which is allow us to provide kind of continuity of teaching. We teach first how to do multi-omics analysis in our course on genomics, and then for imaging informatics course, we teach how to extract features and apply quantitative analysis, very much similar to multi-omics analysis, to image analysis data type. So it's called radiomics analysis.
Are you planning to expand into additional course types in the future?
Theoretical, of course, this platform is universally applicable to any kind of big biomedical data. At this point, we don't really have additional courses where we could implement it. However, we are creative people. We're constantly looking for new opportunities to provide our students with the best experience in training. So I wouldn't exclude the possibility that in the future we'll use it for more courses.
Yeah, I think the Dave's hands-on session on the imaging pipeline was very well received by our students this semester. So next year, we're going to have the same imaging course in the spring. So we will continue to collaborate with both of you and also Dave for the probably a more expanded version of the imaging portion of the imaging part of this SB, perhaps.
Yes, that's a good point. I just want to add that our plans is to expand this imaging informatics course in a full semester course. Right now, we teach it in the summer as a compressed course, 1 month course. So we're planning to expand, and we're definitely planning to expand the topics covered with the workshops on Seven Bridges for imaging informatics to make it a more comprehensive training.
Great. Yeah, and we'll be happy to provide any kind of support and help to make it more and more accessible to the students. So just one more question related to that. I know curriculum development is a lot of work, and I know both of you are deeply involved in that. So you've already explained about how the CGC is helpful in setting up everything. So I just wanted to get some feedback on, like, what do you think will make it even more easy to set up your the course on the platform?
Kritika, do you have any ideas?
Yes. So yeah. So this time when we were doing the course, I had to set up the projects for each of the students. So all the projects are currently under my ownership, but at the end of the semester, we would like to transfer the ownership to the students so they can actually keep a copy of the pipeline and the project under their own credits in case they want to continue like practicing and using it for any of the interviews or any further practice. So I think that would be helpful to have, is the ability to transfer ownership to the students once at the end of the semester. I think would be very helpful. Is that the answer that you were expecting?
Great feedback. This was not, this question was not there, but I just, I felt like it was appropriate to ask you. We don't have to.
Oh yeah, absolutely. And this is actually a very good comment Kritika made. We see that many students are interested to continue using platform after they done with learning in our classroom. And we have some examples where students already using it after they graduated from the program.
Can you go into a little more detail about how the students are using the platform after they take the class?
Right. One of the success stories I would like to share is one of our recent graduates was actively searching for jobs after graduating from our program last year. And he came across a great opportunity to a job opening in a very prestigious medical research organization in the US, where he wanted to be a bioinformatician. During his interview, he actually used a live demo using Seven Bridges account, which he uses during training, and he uses the pipelines he learned during our course on precision health informatics to demonstrate during interview how he can handle genomic data processing on a cloud at Seven Bridges. I know from his feedback that there was a very surprise effect on the interviewers. They react very positively. Were very impressed with his skills and ability to show on the fly how he can use this platform. And he successfully landed the job and now currently working as a clinical bioinformatician at this medical organization. And also I want to add, as far as I know, a couple other graduates were using our platform during their a capstone project, which is a summer internship with industry partners, which we have as a part of our program. We provide students the opportunity to have a real life projects with different kind of industry partners. And two of our students were using metagenomic pipelines on Seven Bridges to implement for COVID-related research, which they were doing during their internship with the company.
That's great. Out of curiosity, have there been uses of the platform for students that are doing academic research projects?
So all the capstone projects are industry, do have an industry mentor and a Georgetown mentor. So they are not academic research projects. They all have one industry mentor and they have one Georgetown mentor.
So you can say they have both sides. They have industrial application with academic component to it.
Great. Yeah, these are the success stories we want to hear. And it really fills my heart to see it has helped someone secure a job. So this is amazing. Thanks for sharing that with us. Okay, so now that we're talking about research projects, Dr. Gusev, you are a co-director of the Informatics Shared Resource for the Lombardi Comprehensive Cancer Center at Georgetown. So can you provide us some background on the core services of that lab? What are the kinds of research you do there?
Right, sure. As I mentioned before, we are not only teaching genomic and bioinformatics and imaging, we're also practitioners. We're actively involved in large-scale research projects, mostly with Lombardi Cancer Center researchers here at Georgetown, where we provide collaborative research services and support on the bioinformatics side for various cancer-related, mostly translational type of projects. Translational means that some findings initially made in the research laboratory setting are being investigated additionally to implement in the future possible clinical applications. I'm co-director and Kritika is a manager of the shared resource, bioinformatics shared resource for Lombardi Cancer Center. We work closely with multiple investigators on campus. In many cases, we develop multi-year collaborations as a part of our services. We mostly work with genomic sequencing data, RNA sequencing and others type of sequencing data, and also provide a lot of so-called downstream analysis at the gene expression level, where we do system biology type of analysis. we have several focus areas in our research. We provide a lot of support for research of GI tract cancers, because this is one of the specialty of our cancer center, as well as breast cancer. And we also work with brain cancer related projects. Outside of Lombardy, we also work with the medical center, Georgetown Medical Center researchers. One of the areas with a success story is Georgetown Transplant Institute, where we work with a group of researchers which help to utilize molecular profiling data to understand the mechanism of transplant rejection in patients. For all of this project, I must mention, we actively using Seven Bridges platform. We counted at this point nearly 50 different projects where we utilize on a regular basis Seven Bridges platform for RNA-seq and whole genome sequencing data analysis. We also do our own research to develop novel methodologies which you can offer through shared resources to our collaborators at Lombardy. And as such, we implemented at Seven Bridges a novel viral genomic platform, which allow everyone to use RNA sequencing, human RNA sequencing data to investigate and quantitate a presence of different kind of viral RNA in tumor samples. This platform, this pipeline was developed by our team and was published a couple years ago in Frontiers in Microbiology. So this is another example where we utilize Seven Bridges platform specifically, not only to do data processing and analysis, but also to develop novel tools.
I also want to add, in addition to RNA-seq and whole genome sequencing that Yuri mentioned, we also have used the Seven Bridges platform for running pipelines for on whole exome sequencing for our collaborators. And also, we also use it for HLA typing for our collaborators. And these are inbuilt pipelines inside the Seven Bridges platform, and we have used them for our research projects.
That's great to hear that it's been so useful for so many different types of research. Can you go into a little bit more detail about how the CGC has helped the core lab work on these pipelines and streamline the process for your collaborators?
Sure. One of the major challenges we faced early on when RNA-seq became very popular is how to handle this enormous amount of data being generated every time when the investigators extract RNA from tumor samples and run multiple samples on a sequencing machine. They generate terabytes of data, and it was very difficult initially for us to find a way to streamline the workflow so that we can actually move the data around and find appropriate platform to lend the data and find the tools to bring them to the same cloud. So after several years of searching, we were happy to find a Seven Bridges platform, and we compared it to others, and we found it very advanced and user-friendly. So this platform helped us to move the data after they're being generated in our local genomic core facilities, move the data to the secured cloud environment at Seven Bridges. It's a cloud-to-cloud transfer at this point from base space cloud to Seven Bridges cloud. And then right there on the spot, we have accessible pipelines, which we can modify or use as it is to process the data on a cloud. In addition, our collaborators found this very useful that they all are included in Seven Bridges projects as collaborators, so they can actually participate and see what's going on, how the analysis is done. Some of them actually wanted even to learn how to use this platform after collaborating with us. We provided training for several faculty on campus on how to use Seven Bridges platform. So in a way, Seven Bridges really provided us opportunity to simplify and streamline this handling of massive amount of genomic data and make it easier, and also provide open access and make it shareable with the collaborators. So there's many aspects which we see as very positive in using Seven Bridges.
Yeah, prior to when we started using Seven Bridges, this was back in 2015. Before we started using the cloud, I remember it took me 3 months to analyze 75 whole genome sequencing samples, and this was on a desktop with 32 GB RAM. And after that, well, there was a project which we used the cloud, the Google Cloud, and this the runtime itself was not that long, but because I had to move the data in and out, and then I had to set up the virtual environment, set up all my scripts, and run all of that took time. And it took me probably about a week to process those samples. And those were clinical depth samples. So they were really big files. And then after that, we started using the Seven Bridges platform. And once the pipeline is set up and tested, it's very easy to run them on a web browser. I can start them in the evening. I've done it on a train through my phone. And next day, I'll come back to the office and it'll all be done.
That's fabulous. I know how hard it is to optimize pipelines, trial and error in
Right.
Local high performance clusters, but you simplified it with the metrics you shared with us. So that's amazing. So are there any success stories in the research front? You've shared a success story of how one of your students secured a job using the Seven Bridges platform, but do you have any success stories that you can share in the research front?
Well, in research, the success is measured by publications, I guess. We have several examples where we published together with our collaborators work on liver cancer, for example. And also, I already mentioned, we published a series of publications, at this point, 3 of them, with the Georgetown Transplant Institute, which is also connected to cancer-related research because the transplants mostly are done on cancer patients. So the transplant research, we really pleased, and we think of it as a success story because we run the RNA-seq analysis on the Seven Bridges platform, and it provides us with endless, basically, resource of novel insights in the mechanism of transplant rejection, and we continue collaboration with this group, and they seem to be very pleased with the outcome.
Yeah. I can also add a story we had back in 2017 when Yuri and I were working on our viral informatics pipeline, the VGen pipeline. At the time that we were working on this pipeline, we came across this blog post on the Seven Bridges newsletter. I think it was by Ronak, who had compared the centrifuge pipeline with the Kraken pipeline, and it was done on Seven Bridges on some specific, I think it was HPV or some specific cancer type, and they had detected a bunch of viruses from those from his example. So we actually contacted the Seven Bridges, and they put us in touch with Ronak, and he helped us to create this VGen pipeline on the Seven Bridges cloud and sort of optimize it to run on the scatter-gather mode on Seven Bridges platform. And we were able to compare the output and results from our pipeline with Centrifuge and Kraken. And that is actually published in our paper. And we have acknowledged Seven Bridges, and we have also cited the blog post by Ronak. So I think that probably gave us an additional dimension to our paper. So it was very helpful to be able to compare these pipelines using Seven Bridges.
Yes, and I just realize I want to mention one more publication. We just posted on bioRxiv a preprint of our paper, which is currently under consideration in a journal, peer-reviewed journal, where we applied this viral RNA pipeline to COVID-19 data from the lung biopsies of the COVID-19 patients, in addition to also applying to the same RNA-seq data the metagenomic pipeline through the 7Bridges platform. And we think this is a very interesting new development where we can actually utilize multiple pipelines on the same platform, multiple pipelines on the same platform to provide this multi-dimensional research of the specific disease domain, in this case, COVID-19 domain, which of course is a very important topic these days.
That's great. It's great that the platform has been so flexible in terms of helping in different subject areas, and in this new emerging subject area, so quickly you've been able to start researching using the CGC. What advice would you give to other researchers that are just getting started with the CGC or working in the cloud in general?
Right, that's a good question. We actually doing this kind of advices regularly for our new coming faculty. Just maybe last week, I have connected a new faculty at Lombardi to Dave Robertson to make it easier for him to start using 7Bridges platform. I always recommend 7Bridges to our cancer researchers because of the simplicity and easy to learn platform, and also availability of very important data sets such as TCGA and others. In cancer research, it's become customary to utilize TCGA collection, for example, to match and compare the results generated by researchers to the publicly available data at TCGA collection. So it's an important part of modern cancer research, and 7Bridges provide easy access to the entire collection of TCGA, as well as target collection and TCIA collection for imaging, which is also emerging and very important part of modern cancer research. The utilization of imaging in a quantitative way together with other omics data.
Yeah, I think the barrier is very low for new researchers to learn and learn to use a 7Bridges platform. Even within our center, we have been able to share our RNA-seq pipeline with our colleagues, and then they have been able to apply the same pipeline on their own data set we just share the pipeline with them, and they're able to run the same pipeline on their own data. So I think, and we also have several training and seminars that Yuri does for researchers in the Lombardi Cancer Center and also Georgetown Medical Center. So I think the barrier is very low because the data, the tools, and everything is already on the cloud. All they need is an account, and it's easy to get started.
Yeah,
with the free $300 credit, so it's not that it's very easy to get started. Yeah.
Those are great points, actually, and that's how we want to empower all our users and our collaborators, and I think that's the most powerful way of promoting the platform, right? Our users doing it among their own, communities. So that's really fabulous. So with that, one last question I wanted to know is, you've already shared a lot about the CGC, how it has been used effectively for the training and also for your academic research. Do you want to share anything else about the CGC?
I have a kind of an idea I want to share, or maybe a suggestion. In the past, there was a very successful program called NIH Credits, Cloud Credits program, which we participated in. That was an NIH-sponsored program, which allowed researchers to obtain to apply for small grants and to obtain access to 7 bridges with credits, certain amount of credits, usually an amount of $10,000, which is plenty to run even a large scale projects. And for academic researchers specifically, for academia, I see this as a very valuable way to make it easier for researchers to use 7 Bridges platform. I wish this program would be resurrected in one form or another, so that at least some academic researchers through NIH-sponsored funding could get access to this platform on a continuous basis. It's very important to realize that once you establish your pipelines and your data sets on 7 Bridges, it's convenient, but it's also practical to continue maintain the presence of the researcher on a platform. And for that, we need continuous funding, obviously. So one way or another, I really would encourage 7 Bridges as a company to continue be very friendly toward academia and provide a low barrier, even a financial low barrier for academia to use this platform, because there is a great benefit for academia and for 7 Bridges in these collaborations.
Yeah, I completely agree. I think there was a conference at the TCJ conference when we went. There were several groups there who had this NIH funding, and it was very interesting to see all the pipelines that they had built through this cloud credits. So I completely agree with Yuri. At least for academic centers, I think this kind of small grants is very valuable.
Yeah, 7 Bridges is very happy to continue to support academics. And you bring up an excellent point that the funding needs to be sustainable. You can't just do a one-time project. Ideally, you would be looking at it the same way you look at consumables in your lab. You can't just get pipette tips once.
Yes, I agree.
And an important point about including it in grants and the importance of sustainable grant funding going forward as well. Well, thank you so much for your time. We really appreciate your sharing your thoughts on how you use the CGC, both in terms of teaching master's students and in terms of your own work in the core lab. So thank you again, and we look forward to working with you on new courses in the future.
Thank you. It's been our pleasure working with you guys and talking to you.
Thank you so much.
Thank you. All right.